Um, Going back to how we measure pH, we have a qualitative way to do it, that is just looking for color changes. We use these things called acid-base indicators. They usually come in these little bottles we use in the laboratory. This is phenolphthalein. You should watch the other video I put on Canvas where I show you the how Le Chatelier's principle can be demonstrated using this indicator, just causing shifts in the pH by adding outside acid or base. This turns pink in base, and it's colorless, like in this bottle, in acid. And it's the reason why indicators change color is because indicators are somewhat complicated looking molecules. Like this is the structure of phenolphthalein. I'm not going to expect you to know how to draw that, don't worry about it. There's another indicator called bromocresol green, but you have a variety of acid-base indicators to choose from. They're usually plant extracts, dyes that are found in cabbage, red cabbage, or tomatoes, things like that. But the reason why, for instance, phenolphthalein changes color is because in acid it has some structure, usually from a pH from 0 to 8.2. So on the acidic side of things, it has this structure. When you add outside base to a solution that contains that indicator, the indicator is going to change color and lose 2 of its protons, and it shifts its structure into something like this. In this structure here, it actually is able to absorb visible light. This structure does not have the right energies to absorb visible light, but this one does for reasons we won't worry about. It has something to do with these alternating double and single bonds. But this particular arrangement of electrons causes it to absorb light. Now, phenolphthalein just happens to absorb greenish light best, which has a wavelength around 553 nanometers. That just happens to be a greenish color. If you look at a color wheel, this is just like a basic color theory. A color that you see with your eyes is always the complementary color of what's absorbed. So if this particular structure of phenolphthalein that's present when the pH is above 8.2, then it's going to, if it absorbs green light, the complementary color of green is red. That's why it has a pinkish color. It's not exactly a red color, but it's in the red part of the color wheel. So look at that other video to see the color changes I'm talking about. It's on Canvas as well. But that change occurs at 8.2 pH or above, so on the basic side. So this particular indicator is useful for indicating the endpoint of a strong acid, strong base titration. The next lab that I'm going to post over the weekend, it will be dealing with that sort of reaction, and I'll be using this indicator to detect the what's called the equivalence point or the end point. There's another indicator. They all don't have that. They have various colors. This indicator, called bromocresol green, has very pretty colors. It turns this yellowish color in its acid form at low pH. At a higher pH, it changes its chemical structure and absorbs or looks blue to us. So this is how we use them. It's not an exact estimate of pH. It's just to tell us that the pH is changing. That's a very common color change there. Blue to yellow, yellow to blue. We can also use indicator paper. It turns different colors depending on the pH. This particular paper turns very bright red when it's in acidic solution, and it's in water, it's just a neutral green. And in very basic solution, it turns a very almost hunter green, dark green, blue color to indicate that the pH is close to 14. We can also use litmus paper. Litmus paper is that stuff you probably used in the lab before. It is a plant extract that is red when it's in acidic solution and blue when it's in basic solution. So what they do is they coat paper with this dye, And if it's in the blue form and it turns red, that means that you have an acidic solution. The reddish paper will turn blue when it's in base. So it's either going from left to right or right to left to indicate that you're either an acid or a base. But again, that's only a rough estimate, qualitative way of explaining or detecting what the pH is. A more exact way to do it is you use something like this. This is something you can connect to a computer. It's called a pH probe or pH meter. It's just a long glass tube. You can only see the very end of it. It's very delicate. That's why they have this plastic around it. But that tube you see there, that bulb you see right there, is a very thin glass bulb that allows H plus ions to go through the glass. It's a semi-permeable membrane. And depending on the solution you put this in, different amounts of protons will be able to go across or go through that bulb, and that's converted into a voltage, and that will tell you the pH of the solution. That's a very common way of measuring pH in a solution. Our last topic is buffers. Very important topic to have a basic understanding of, and that's what our last couple slides will be discussing here. So I want you to be able to recognize what a buffer, when you see a buffer solution, and why it resists changes in pH. The verb to buffer just means to lessen or to moderate. So buffer solutions in chemistry resist changes in pH. They're very important. They keep the pH of our blood constant. If we didn't have buffers in our blood like this, the bicarbonate carbonic acid buffer is the main one in our blood. If we didn't have them, we wouldn't survive very long, because when we exhale or inhale air, we're changing the amount of CO2 in our blood, and CO2, when it dissolves in water, forms carbonic acid, and that will lower the pH to a dangerous level if we didn't have this to counteract that. And that's what a buffer is. A buffer is simply a weak acid and its conjugate base present in equal amounts. That's what the best buffers have, these two components present in about the same amounts. And what's interesting is you cannot have a strong acid and a strong base mixture be a buffer. The reason why strong acids and strong bases cannot be buffers is because they will react with each other and neutralize each other, and all you'd be left with would be a solution of water. These weak acids and their conjugate bases, whether it's acetic acid and sodium acetate, or hydrofluoric acid and sodium fluoride, or carbonic acid and potassium bicarbonate, or ammonia and ammonium chloride, these are all types of buffer solutions. The reason why these can coexist in solution is because they're weak acids and weak bases. They don't react with each other. They just develop an equilibrium with each other and are present in about the same amount. So how does it work, chemically speaking? Well, I've drawn a couple pictures here for you. On the left side, we have a buffer mixture. Notice I have a couple reactions written here. This would be the structure of acetic acid, and it's a weak acid, so it develops this equilibrium with its conjugate base. So it does produce a few protons in solution, but a very small amount of them. And it also has about an equal amount of its sodium salt, in this case sodium acetate. All the sodium acetate does when you dissolve it in water is it dissolves and it makes a bunch of acetate ions and some sodium ions. The sodium ions just float around. They don't do anything. They're called a spectator ion. And there's so few protons in the solution that we can kind of ignore them. So really, the only thing that's present in this solution besides water that's important are you have a lot of acetic acid molecules and you have a lot of the acetic or sodium or the acetate conjugate base of that. And over here on the right, imagine you have just a beaker filled with pure water. At this point, there's no hydrogen chloride, no hydrochloric acid added. So what happens when you add concentrated hydrochloric acid to both solutions? Well, if you just add HCl to water, all that's going to happen is the pH is going to drop very quickly. The reason is because water has no ability to resist changes in pH. If you add HCl to the pure water, all that happens is the HCl completely breaks apart into protons and chloride ions. So you get a pretty quickly a large concentration of protons in the solution. As the hydrogen ion concentration goes up, as you know, the pH of the solution drops. So after you add the HCl, the pH will be much less than 7. So it's a very acidic solution. Contrast that with what would happen when you add HCl to a buffer solution. When you add HCl to a buffer, the buffer has a base, a weak base, that will neutralize the added HCl. So this reaction would occur here. HCl will react with the large amount of acetate ions that are already there in the solution, and the acetate ions completely react with the HCl. Because this is such a strong acid, the reaction is driven to the right, and those are completely consumed, but they neutralize the HCl to just make more of the acetic acid and make chloride ions, which are spectator ions. They don't do anything to pH. So all that you're doing is converting the conjugate base that was already there in the solution to its parent acid, which is already there in the solution. As long as you don't add too much HCl, the amounts of these don't change. This stays pretty constant and that stays pretty constant. So it's just like a vacuum cleaner for the protons. They get sucked up and neutralized by the component, in this case the basic component of the buffer. So the pH does not change. There's no proton. The key point here is that there's no protons or hydroxide ions generated in this situation. They're completely neutralized. So therefore the pH does not change for a while at least. Eventually you could overwhelm the buffer and add in so much HCl that eventually the HCl will neutralize all these, and all you'll be left with will be a solution of hydrogen ions and acetic acid, so the pH would quickly drop. But we're talking about situations where the amount of added acid or base is not too much, so the buffer has a certain, what we call, capacity. It has a certain buffer capacity that resists these changes of added acid or base. Now if you add base to this mixture, if you added NaOH, all that would happen would be this reaction here. This reaction would occur. So I'll review that in a second. Let me just go back to what's going on in this mixture. There's an equilibrium going on. You have equal amounts of acetate and acetic acid, and this argument applies to any buffer. You just have to be able to specify the acid and the conjugate base correctly. The conjugate base, notice, is always negatively charged, and the parent acid is neutral. So again, this is the major equilibrium that's set up within the buffer. The weak acid dissociating to give you conjugate base and a few protons. As I just showed you, if you add strong acid to a buffer, in this case, let's say you add HNO3, there's so much acetate floating around from the buffer solution that it will completely neutralize any added nitric acid. It'll consume the protons and just be converted into its parent acid form. The conjugate base of nitric acid is nitrate ion. It's a very weak base, essentially has no effect on the pH. So now you still have these are gone now, or that's gone, that doesn't do anything to pH. So you have these two present, and the pH of that after you add the acid is pretty much the same as the pH before you added the acid, because they were already there. One buffer component is converted to the other. That's the key. If you add base to this mixture, all that happens is instead of the conjugate acid of the weak acid taking care of the added acid, you have the acetic acid itself reacting with any added hydroxide ion, strong base. OH reacts with a proton here to make a water molecule, and once this loses a proton, it's converted into its conjugate base. So you get sodium acetate and water. So again, any added H plus is neutralized by the base of the buffer. Any added base, OH minus, is neutralized by the parent acid of the buffer to convert into the opposite or the other piece of the buffer system. That's how buffers resist changes to pH. They destroy any outside added acid or base. So that'll finish our discussion of acids and bases. I know that's a lot of material. So you have 3 relatively long videos, and this 4th video is about 15 minutes. Please watch them carefully. You're going to have an online quiz that I'll post over the weekend. It'll be you'll have about 3 days, a 3-day period to take it. And then again, I'm working on that lab to post over the weekend that will be due later next week. So with buffers, really what will you be responsible for? Well, you should be able to recognize when you have a buffer and be able to write the chemical equilibrium for what's going on if I give you a buffer solution, and to explain or to understand why it resists changes in pH. So just to finish up, let me just give you another example. It always helps to visualize it for you. So let me find a piece of paper here. Maybe I can just do it on the other side of this piece of paper here. Let's say you had this buffer. Let's take a simple one, this one here. So let's say you have a beaker filled with, how do you make a buffer solution with these 2 components? Well, you'd add in equal amounts. So a good buffer would be like, let's say, 1/10 of a molar solution of hydrofluoric acid. Now that is not a strong acid. That's the one halogen-containing acid that's a weak acid. And a good buffer would contain about the same amount of its sodium salt. Why sodium salt? It doesn't matter. It could be the potassium salt or the lithium salt. What that does in solution is it breaks apart into sodium ions and fluoride ions, about 0.1 molar of each of them. So this is a good buffer solution. So it has a weak acid and it has its conjugate base. The sodium ions don't do anything. They just float around and watch the action. So let's say you add in some HCl to this buffer. How would you explain how this buffer system resists changes in pH and keeps it constant? Well, you'd say if this is an acid, it's going to react with a base. The only thing in this solution that's a strong enough base or a base of any kind is the fluoride ion. Remember, in these solutions, there's always water. But water only very few of them exist as H plus and OH minus. So essentially that's not going to do anything, but this will. Fluoride ion will neutralize or react with HCl in this reaction. It's a complete reaction left to right, and you just make hydrofluoric acid and you get the conjugate base of the hydrochloric acid. So all you're doing is converting one part of the buffer into another. So the change in pH is essentially 0. It resists changes. If you add in hydroxide ions, and let's just say you add in KOH, what happens? Well, you should be able to recognize that that will be neutralized. So the reaction that would occur would be KOH would be neutralized by the acidic part of the buffer, hydrofluoric acid. And that's a complete reaction. That would form a water molecule and potassium fluoride. And note, the key thing to remember is even if you add acid, that's all going to be consumed. A small amount of acid will be completely consumed, and you're still left with an equilibrium mixture that you had before you added the acid. Same thing here. Add in base. As long as it's not too much base, it's completely neutralized by the added acid. So after the reaction occurs, all you have is a little bit more of the parent weak acid and a little bit of its conjugate base, because potassium fluoride is just a soluble salt, and it's potassium ions and fluoride ions. So there's your conjugate base of the weak acid. So that's essentially how buffers work again. like vacuum cleaners for added H plus or OH minus. So recognize that when I add that, I'm really adding in a source of H plus, and when I add that, I'm really adding in a source of hydroxide. Those are the active parts of a base and an acid. That K plus or the sodium ions don't do anything in the solution. So again, good luck with everything. Just keep working hard, and we will talk to you next week. The next topic we're going to go into after we finish the lab for acids and bases will be organic chemistry, which we will get into mid next week.